So, what I want to do tonight for the next hour or so is I want to tell a story, is really what I want to do. And I want to share this story with you because my main interest in all of this world of fungi really is the evolution of co-adapted species. And so, as species evolve, they change each other in a feedback loop that causes them to evolve in different directions, but also it causes them to affect each other in ways that's fun to try to unravel the story of that. So tonight I want to tell you the story of decomposition or saprobe nutrition in kingdom fungi. So I'm going to jump right in here and start by saying that these are the three stories that we see most often when we look a little bit past identification and edibility and we start investigating the ecology of fungi. And the first big story is really the story of pathogens. And a good example of that is the chestnut blight, in which a fungus killed all the American chestnut trees for the most part in North America. That was an introduced fungus. And also, likewise, Dutch elm disease, another introduced fungus that was introduced to North America on a beetle, carried by a beetle that killed America's favorite shade tree, one of the most popular cityscape trees. And then the other pathogen or parasite story that we all know and love is the story of cordyceps and the zombie cordyceps, and of course, the television series The Last of Us. So that's one big story that we all have been following. The other big story is the story of mycorrhizal symbiosis, and that's a story that everybody knows, the story of the Wood Wide Web. And everybody has probably heard of Susan Samar's book and videos, Finding the Mother Tree. And if you haven't heard of the Wood Wide Web, there are about 10,000 podcasts and YouTube videos about that subject. And in fact, that subject got a little bit carried away from the science part of it and sort of drifted over into the hyperbole uh, range of storytelling. And so, what we want to do is acknowledge that, maybe rein that story in a little bit. And tonight, the third big story is a story I want to talk to you tonight about, and that's the story of decay and recycling, or the nutritional mode of fungi as saprobes, or organisms that can decay organic material. So that's where we're going tonight. And I want to ask the question, how do we tell these stories, science stories? And so one way we can tell the story of the saprobic lifestyle is through science, and another way is through narrative. These are two approaches to communicate what we know or what we think we know or what we have learned so far about the story about how fungi obtain their nutrition. And so that's what I want to do tonight. I want to tell that story, even though I know that everybody here already knows that story. And so, what I want to do is, I want to communicate the story of decay and decomposition in plain language. But I want to stay true to the science while we do that. So, it's important that we do that as communicators, as educators. We communicate in regular language, but stay true to the science to the best of our ability. So, how do we learn? What are the ways we learn? So, to me, this is what science looks like. You can see that the scientists are all geared up in their scientific gear. Someone is taking down data, someone is doing chemistry, someone is looking through a microscope. And if we were to report a scientific report of the fungal nutritional mode of saprotrophy, you would see a lot of charts and graphs in bioinformatics that describe data trends and interpreted those data trends. I'm not going to do that tonight. What I'm going to do is what you see in the next photograph. I am going to tell a story. And the story is plain language and easily communicated, easily understood. And we're going to try to get into some kind of complex chemistry in regular plain language. So, narrative probably began in Paleolithic human terms around a fireplace, telling stories about the hunt or telling stories about which foods are edible, or telling stories about which foods or fungi might have medicinal values. And so, narrative has always been a way to communicate the way we investigate the world. And that's what you're going to get from me tonight is a narrative story. So, welcome to the Wood Wide Web. I know what you're thinking. I thought I was going to talk about decay, and I am going to talk about decay, but I want to talk about decay from the point of view of the other Wood Wide Web, and that's this Wood Wide Web that you see over here on the right, that mycelium that is in the forest underneath the leaf litter and underneath the pine needle litter, and it's crawling around between the soil and the forest floor litter, and it's searching for a new substrate to colonize. And so, what is that? That's not the Wood Wide Web, it's the Wood Wide Decay Web. So we all know the story of the Wood Wide Web as a mycorrhizal symbiosis story, and I'm going to tell you the story of the Wood Wide Decay Web, and that is when fungi are digesting a log or leaf litter or branches, and they start to exhaust the nutrients, they are going to leave that log and go out in the forest and search for a new substrate to colonize. All right. So when Copilot and AI first became available to the public for free, I asked Copilot to draw me a picture of what the Wood Wide Decay Web might look like. And this is the picture I got. And you can see that's pretty weird any way you look at it. They had the idea in general of mycelium crawling out from underneath the litter and the moss, but it looks to me like they have the mycelium attacking honey mushrooms. And that's not really what I want. So what I did was I went to Chat GPT, the first version, and I said, please write me a story about mycelium that has left a colonized log because it was running out of food and it's searching in the forest for a new substrate to colonize. And I'm not going to read the whole thing, but I'm just going to tell you that even in the first weeks of ChatGPT, ChatGPT did an excellent job answering my question and crafting a story about mycelium crawling around on the forest floor looking for a new substrate. And I want you to read along with me the third paragraph where it says, my, first of all, they named the mycelium mycos. I thought that was cool. Little mycos mycelium. Anyway, Mycos is crawling around and he's having adventures. And then it says Mycos encountered a variety of unique substrates on his forest adventure, each requiring a new set of challenges to his enzymatic toolkit. But eventually, the mycelium was able to thrive, absorbing nutrients and decomposing organic matter along the way. Well, I thought that was really excellent. Excellent. for artificial intelligence because it actually captures the essence of what really happens when mycelium is in search of a new substrate to colonize. All right, before we go too much farther, I want to just go back over what I know everybody already knows. So fungi are the primary decomposers and recyclers on our planet. That's pretty much evident. I'm sure everyone knows that. Bacteria do a little bit of these things, but fungi are the main players in decomposition. And by doing that, what they do is they drive all the main geochemical cycles that support life on the planet, and that includes the carbon cycle, the nitrogen cycle, the phosphorus cycle, and in some ways also the water cycle. So those cycles that make life possible by recycling nutrients, essential nutrients, are pretty much driven by fungi decomposing organic material. And the way they can do that is because fungi have unique chemical abilities. And everybody who ever talks about fungi as an educator, what you're going to do is you're going to tell people that there's two main kinds of decomposition of woody material, and that's white rot and brown rot. And we're going to get into a little bit of that. And I want you to get familiar with the details of white rot and brown rot so you can explain it to people that don't have any background in chemistry or biology or mycology, and just increase your own understanding of how those different kinds of rot function. All right, so the fungal lifestyle of decay and decomposition and rot. And this is my disclaimer slide. This presentation explores the saprobic mode of nutrition. And what we want to do is understand that that nutritional mode is called external digestion, and food or organic materials are digested outside the body of the fungus, and they are absorbed through the cell wall. And so in ecosystems, fungi break down wood, forest debris, all organic materials, and it frees up the nutrients for everything else. And I cannot take credit for any of the science in this program I'm sharing with you tonight. This program is all based on the work of actual real scientists, mycologists, Lynn Bodie, David Hibbett, Francis Martin, Jonathan Platt, Tom Sprung, and more than anything else, the 1000 Genome Project. And that's from the website Microcosmos, in which a consortium of mycologists from all over the world are working together to do a full genome sequence of 1000 different fungi. Starting with Lacaria bicolor, that was the first one, and then the goal is to sequence 1000 fungi with the whole genome. So a little backstory, and so we want to talk a little bit about the different modes of nutrition in fungi. First, we have biotropes, and those are the parasites. Cordyceps is a good example, or pathogens, chestnut blight. And then we have mutualists. Then we have all our ectomycorrhizal and our buscular mycorrhizal, all 7 different categories of mycorrhizal symbiosis. And then we have the necrotropes or the saprobes, and those are the fungi that feed on things that are already dead. And we have leaf litter and needle duff decomposers, and then we have the wood decomposers, the white rotters, and the brown rotters. So that's sort of an outline of the different modes of nutrition. I want to spend a little minute on this flow chart here. These are the 5 modes of nutrition among fungi. And I just want to make the point here that they are all interconnected and they all inform the lifestyles of each other. If you start with the parasites, and you look at the parasites, lots of parasites, if you follow this arrow here, if you can see my cursor, lots of parasites begin as a parasite and then they switch over to mycorrhizal symbiosis. And also, several parasites can also switch over and become a white rot fungus, or they can start out as a parasite and become a brown rot fungus. And also, parasites can also switch over and as parasites of leaves and needles on living trees, they can kill those leaves and needles, and then they fall to the ground, and then those parasites will switch over and they will become a decomposer in the leaf litter or the needle litter. If you look at mycorrhizal symbiosis, there's only one arrow that goes from this red bubble here over to the leaf litter. And, and that's because sometimes mycorrhizal symbiosis doesn't have the energy supplied from a tree to the fungus to complete the job of allowing the fungus to form a fruit body. So a lot of times ectomycorrhizal mushrooms like say boletes or amanita, they leave the colonized tree root on their way to the surface to form a mushroom, but when they get to the surface, the tree has not supplied enough energy to the mushroom, so the fungus switches over to becoming a litter decomposer to get that last bit of energy to form fruit bodies. And I'll talk about that again later on when I talk about why uh, ectomycorrhizal fungi form fairy rings. Because if you think about it, that not really doesn't make a lot of sense. But we'll get back to that. And then over here in the needle leaf litter bubble here, you have an arrow that goes over to, whoops, to wood decay. Let me move back there for a minute. And then number 4, brown rot. You can see that brown rot evolved over time into ectomycorrhizal fungi. All of the boletales, all the boletes, and everything in the bolete lineage, at least up to the order level of boletales, evolved from brown rot fungi. And also white rot fungi are capable also of evolving into ectomycorrhizal fungi. And white rot evolved into brown rot about 180 million years ago. I know that was a lot, uh, and because this is complex, it's not complicated, but it's complex. I wanted to get that in your head and swimming around in your head, how all these different nutritional modes are not static. They move back and forth among various members in all ecosystems. But let's get back to decomposition. Decomposition is what we're going to concentrate on here, or saprotrophy. And at least part of the life of all fungi has some part of saprotrophic mode. And that process is pretty much the same always, where the hyphae penetrates the substrate and releases enzymes that break down large organic molecules into smaller subunits, and they diffuse back into the hyphae through the hyphal cell wall. And there's different enzymes involved in all these different substrates, thousands and thousands of different enzymes. All right. This is my drawing, and I stole this drawing from Jens Peterson, or borrowed this drawing rather. And what I want to do is just go through a little bit of your standard decomposition routine. So my cursor is on a single hyphae, and at the end of this hyphae right here is where all the action takes place. The very tip of a hyphae is where the enzymes are extruded or excreted into the substrate. And in this drawing, we're on a piece of wood, and the hyphae is lying on that piece of wood. And these are enzymes that the fungus just excreted from its tip. And the fancy name is a lignocellulitic enzyme. That's not important. It's a magic chemical. And what the enzymes do is they attach to woody molecules, in this case, where the cursor is now, a molecule of cellulose. And cellulose is a chain of simple sugars. And so what the enzymes do is they break the bonds that hold the individual simple sugars in the chain apart. And so what you end up with are just molecules of glucose from the cellulose, and then the glucose can diffuse back into the hyphal tube of the fungus body. So if I get to a point where I'm too excited and you're not following what I'm going to say, uh, I'm going to ask one of the moderators if they can stop me. And if someone has a question, we'll stop for a minute. But let's move on anyway. This is a close-up of what you saw. And so now we're back at the tip of the hyphae right here. And I want to talk about this because this is one of the coolest things in all of the ecology of decay. This area right here has a big fancy German name. It's the Spitzenkorfer. And the Spitzenkorfer is sort of like the command center where the nucleus of the fungal hyphae decides what chemicals it needs to decay whatever the substrate is. So the Spitzenkorfer is organizing the creation of these little vesicles. Whoops. Sorry about that. I went the wrong way. All right, I'm back. Is everybody still there? Did you all take a break? All right, so the Spitzenkorfer organizes these vesicles full of enzymes. And the cool thing about the Spitzenkorfer is it organizes the vesicles enzymes for three purposes. Some of these little vesicles are going to excrete an enzyme that dissolves the cell wall at the tip of the hyphae. And some of these little vesicles are going to have enzymes that go out into the substrate. Here, more of those lignocellulases that are going to decompose cellulose and hemicellulose and even lignin to make it into small sugar molecules to absorb through the cell wall into the hyphae to feed the fungus. And some of these little vesicles have different enzymes that are going to reconstruct the tip of the hyphae a little bit farther ahead of where it was before it excreted the digestive enzymes. So this is how the fungi moves forward and excretes digestive enzymes all at the same time and grows through its food, and that's how it travels. So a Spitzenkorfer is something everybody should put that in their vocabulary list. Spitzenkorfer is one of the coolest things in all of nature. It's a command center, and it uses ribosomes to make prescription enzymes for all these different purposes simultaneously and constantly. All right. All right. I'm excited. I hope you guys are too. So I want to slow down a minute and I want to tell you Tom Brunn's story of decay or the saprobic lifestyle. And he makes it really, really simple. And you could tell this story this simply. And it works like this. What is sugar? Uh, when I say that, what I mean is wood is made of cellulose, and that's a structural carbohydrate, and a structural carbohydrate is a sugar. It's a sugar chain. Wood is made of hemicellulose, and that's another structural carbohydrate, and that's another bunch of sugars that are linked together. And then wood is also bound together by a substance called lignin. And lignin is a complex molecule of three different kinds of alcohols that the fungus has to deconstruct in order to get at the cellulose and at the hemicellulose. So Tom Brun says this: what is sugar? Fungi like sugar. Fungi have magic chemicals that transfer wood into simple sugars. Fungi eat sugars. That's the whole story of what we're talking about tonight. Uh, I know I'm making it sound more complex than that, but at the base, at the very base, that's what this story is. And that's what's going on, really. And so, what I want to do is talk just for a minute about what wood is and what those magic chemicals are. So, a little bit of vocabulary, don't worry about this, but the four molecules of life, all life is made of these categories of organic molecules: carbohydrates, proteins, fats, and nucleic acid. And a polymer is just a chain of monomers. And so monomers are building blocks that build polymers. An example of how that works is the carbohydrate is just a chain of simple sugars. And glucose is the monomer, and then that chain of carbohydrates is the polymer. And it's just kind of useful to know that that's what's going on with in the chemistry of organic chemistry of the biopolymers of life. So everybody knows what wood is. Everybody has wooden furniture, I think, and everybody knows about trees. And so these different parts of wood all have different combinations of those elements, cellulose, hemicellulose, and lignin, in their construction. And then a crosscut of wood like that gives us an opportunity to look at wood in the way it's constructed in a tree. And I'm going to talk about this some more. I'm not going to dwell on this slide, but the heartwood of wood is mostly dead cells, and it has low moisture content. The sapwood of wood has live cells, and it conducts water, and it has a high uh, moisture content. And so that keeps fungi out. The high moisture content of sapwood uh, corresponds to low oxygen, and so that keeps fungi out of wood. And so sapwood and bark are both mechanical barriers that exclude fungi from entering healthy wood. And so what is wood? It's the structural material that allows trees to get tall. Cellulose, hemicellulose, and lignin. Lignin is the craziest molecule that I've ever come across, and it has a very complex way of forming, and there's possibly 17 billion different isomers of lignin, which simply means that every time lignin is formed in any plant, anywhere, it's formed a little bit differently than every other plant. And it's still lignin, but it's just a different form of the lignin. And that's why lignin is so difficult to decompose, and only fungi can decompose lignin. All right, so that's a little bit about wood. I want to talk a little bit about the magic chemicals. I don't want to get hung up on the names of these. I'm going to show you the complicated chemical names of these one time, but we're just going to call them magic chemicals. So lignocellulases is a term for all those enzymes that can decompose wood, and it's mostly the chemicals of white rot fungus. Class 2 peroxidases are a class of chemicals in the lignocellulases, and they specialize in breaking down lignin in white rot fungi. And Kzymes is just a fancy word for carbohydrate active enzymes. And those are the enzymes that break down cellulose and hemicellulose, particularly. And this is the biggest class of enzymes in fungi, plant cell wall degrading enzymes. And those are just terms to categorize the different kinds of enzymes. And then in a little bit, I want to talk about this incredible invention of fungi, which is brown rot fungi. And they invented this chemistry called Fenton chemistry, or the Fenton pathway, in which they figured out how to depolymerize lignin without using enzymes. And the way they do that is with free radicals, radical species of oxygen that basically smash apart the lignin momentarily, and that allows little tiny small molecular weight enzymes to enter into the wood and dissolve the cellulose and the hemicellulose before the lignin reforms. And that's a crazy story in itself, how lignin gets unzipped or smashed apart by Fenton radicals, and then it quickly reforms. All right. So I don't get hung up on that this is chemistry and it's complicated. It's not important. What's important is that we're exploring the magic chemicals of fungi and how they take big molecules apart. That's really all we're doing. So in order to rebuild the world, we have to deconstruct it. And that's what's happening with wood, white rot and brown rot. And there is something called the gray rot. And then there are other kinds or ways in which scientists describe rot. A dry rot and a wet rot are two different kinds of brown rot. And it has to do whether or not the enzymes use water or produce water as they break apart the molecules. And then where rot occurs also has its own set of categorical names. Heart rot is when the center of the tree, the heartwood, is rotten. Butt rot and root rot is when the tree rots right where the stem meets the ground and the roots begin. And this is a pathway. Butt rot, root rot is a pathway for heart rot. So lots of fungi enter the tree at the butt of the tree, right where the stem meets the ground, and then they work their way into the root, and then they get into the heartwood of the root, and then they travel up the tree through the heart rot because remember from the previous slide, there's low moisture in the heartwood and the cells are all dead. And then another kind of rot we all encounter is leaf litter and needle rot. And then pocket rot is a kind of white rot in which the tree that's attacked sequesters or encapsulates the rot so the rot will not spread. But it's all just a lot of rot. All right. Well, I'm going to take a deep breath now, and then I'm going to talk a little bit about white rot and brown rot and gray rot. So on the left, you can see brown rot. This is what people call a stringy brown rot. And that happens when a fungus like turkey tail, which is a white rot fungus, it decomposes cellulose, hemicellulose, and lignin faster than it decomposes cellulose. So the turkey tail fungus has to decompose all three parts, the lignin, the hemicellulose, and the cellulose, but it decomposes the lignin first and then the hemicellulose. And then finally, the last thing is the good stuff, the cellulose. So when you see a brown rot, a white rot fungus, and you break that log apart or that branch apart, it's white like this because that's mostly bleached cellulose. All right, here's a couple examples of a white rot fungus oyster mushroom, which is a white rot fungus of hardwood trees. On the right, funeral bells of the deadly Galerina, a white rot fungus mostly of conifers. And here's a white rot fungus of leaf litter. That's on the left, Marasmius strictipes. On the right, here's a white rot decay fungus of pine needle duff, and that's Rhodocolibia maculata. So those are just examples of the different kinds of white rot. And white rot can occur in all different parts of the tree. As a pocket rot that's encapsulated by the tree, as a root rot or a butt rot, as a heart rot, uh, wood chips and mulch, and white rot is really the most common rot. It's also the oldest of all the rots. And so what we're talking about when we talk about the position of decay is this. Decay can occur anywhere on the tree, in the heartwood, in the sapwood. It can occur on the bark. It can occur on the roots or the butt or the stem or the trunk, and even up on the top of the tree. All those are places on a tree that are common sites of different kinds of rot. On the left over here, what you see is a tree branch, and it's infected with a rot, and the rot is rotting the outer part of the sapwood. You can see all that here. The tree is trying to stop, is not quite dead yet. It's probably almost dead. It's trying to encapsulate or stop that rot from spreading throughout the sapwood. And so what it's doing is it's producing chemicals, extractive phenolic chemicals, and here it's starting to encapsulate that rot. So this is what we call pocket rot, where the tree is starting to become successful at encapsulating that rot so it doesn't spread throughout the tree. It's a pretty cool thing. And here's an example also of heart rot. All right, brown rot is something completely different, and we call it cuboidal brown rot because what happens with brown rot is the cellulose and the hemicellulose are decayed, and what they leave behind is this brown stuff, and the brown stuff is lignin. And it's not the same lignin that was in the living tree. In brown rot, this chemistry that fungi invented, the Fenton pathway of chemistry, it deconstructs the lignin so it can get at the cellulose and the hemicellulose, and then immediately the lignin reforms. And remember, I said that there's 17 billion polymers of lignin. When the lignin reforms, it's different than it was when it was deconstructed. And this stuff, the lignin from brown rot, persists in the soil for hundreds of years. It can be a dozen years. It can be 100 years. Some people think it can stay in the soil for 1,000 years. And it's what gives soil, forest soils, their texture and their ability to absorb. And it's a major storage of carbon in forest ecosystems. This residue of lignin from wood decay is the main storage factor for forest carbon in a healthy, functioning forest. I was glad to see Tapenella atratomentosa in the virtual ID earlier. I love that fungus. Uh, and this is a typical basal member in the lineage of the Bolatales. And this is a brown rot decomposer guild fungus that's really the beginning of the lineage of boletes. And one of my favorite subjects is the evolution of ectomycorrhizal symbiosis, and this is part of that story. I want to show you some more brown rotters. So our birch polypore, Fomitopsis betulina, really cool on birch trees. That's a heart rot. That's a brown rot, heart rot, birch trees. And Fomitopsis muncie, what we used to call the red belted polypore, We used to call that Fomitopsis pinicola, but we find out that that's probably not in North America. And so this is the common pine red-belted polypore, and that's a classic example of a brown rot polypore. All right, so white rot versus brown rot in plain language. White rot uses big magic chemicals, those peroxidases, to break apart and digest all parts of wood. Cellulose, hemicellulose, and lignin. And brown rot and white rot is more than 300 million years old. It probably started with chytrids closer to 400 million years ago, and then gradually changed over time. And the modern version of white rot probably arose around 300 million years ago. Brown rot does not use those same exoenzymes as white rot. It uses that Fenton chemistry I was talking about. And what it does is it uses oxygen radicals to loosen up the lignin so little tiny low molecular weight enzymes can get into the wood and digest the cellulose and the hemicellulose and they don't waste energy trying to digest lignin. What happens with white rot fungi is they use so much energy making the chemicals to digest lignin that they barely get any benefit from digesting wood. The benefit of digesting wood, if you have to use enzymes to digest lignin before you can get at the cellulose, it's so energy intensive that it's not really worthwhile. So brown rot is this incredible invention that makes that not necessary. But brown rot's still in the minority. All right, so let's compare and despair white rot. In the white rot pathway, lignin and hemicellulose are decayed first, and that leaves behind some white cellulose. And it's white because it's bleached by oxidation. Well, my screen's too sensitive. And then the remains in a decay log are white and whiter as decay progresses. And it's mostly bleached cellulose because the cellulose remains intact until the latest stages of decay. On the other side, brown rot, the hemicellulose is removed first. And one of the byproducts of that chemistry is hydrogen peroxide. And then the hydrogen peroxide is used in that Fenton chemistry that I talked about that creates radical hydroxyl radicals, which creates free oxygen species. And that's what smashes apart the lignin and it gives the fungus access to the cellulose. All right. I feel like I'm getting a little bit frenetic, but if anybody if anybody wants to ask any questions, anytime you can slow me down, calm me down if you can. But I'm not easy to calm down once I start talking about fungi. So I want to talk about the common fungi that we see, you probably recognize this as the Daleidopsis confragosa white rot. And then over here, something that we used to call the Dahlia quercina, the oak maize polypore. But recently, uh, people have studied the oak maize and Fomitopsis in general. And so we're now going to treat this as a Fomitopsis because it's a brown rot fungus. Fomitopsis is the brown rot, heart rot of oak trees, and all of the Fomitopsis are brown rotters. And so I think it's a good idea to keep the Dahlia in Fomitopsis with the other brown rotters. And so what is gray rot? Gray rot is sort of a combination of white rot and brown rot, and it uses a kind of Fenton-like chemistry that's not exactly the same as Fenton chemistry. And a good example of that is schizophyllum. That's something that has a really complex chemistry that uses a non-enzymatic path that is Fenton-like, but not exactly Fenton, and it also uses enzymes. So that's a good combination. All right. So I want to talk a little bit about endophytes. Endophytes are sort of a hot topic in mycology now. And lots of the earliest colonists that we see on deadwood are probably endophytes. And so a good example of those kinds of fungi are turkey tail and trichaptum, the two that you see right here. And the idea here is that turkey tail and trichaptum and lots of those early colonists live inside of the living tree undetected. There we call them endophytes. And endophytes have all kinds of purposes. Some of them are just lying in wait. They're waiting for the tree to die, or they're waiting for the branch to get broken off in a hurricane. And then as soon as that happens, the endophytes respond as the tree dies or the branch gets broken off, it dries. And then when the moisture content drops, the endophyte wakes up. And so those fungi, the turkey tail, the trichaptum, other pioneer colonist endophytes, they're the first ones on the scene. And Lynn Bodie's name for those endophytes that are hiding inside of living trees, waiting for the tree to die. She calls them the decayers in waiting. And no one really knows how many of these there are. There's probably thousands of them. This is a new sort of field, discovering endophytes, and it has a lot of purposes because endophytes have interesting chemistries in their own right that they utilize as they're lying in wait. And the short and long story of endophytes is that plants and trees, they have their own fungal microbiome. And we're just starting to learn about this. And so they're in a resting stage for the most part, and then they wake up and they function as a saprobe, and they have this home field advantage. And what happens is if they uh, have the first attack on a piece of dead wood, they get some good nutrition, but they are not good at fighting off secondary colonists. So what happens is after a little while, these pioneer colonists get replaced by a second wave of colonists that are more aggressive and take over. We'll talk about that in a little bit. And in the tropics, one of the things that people that study xyleriales, all the uh, xylaria-like ascomycetes, they're in the leaves of all the trees inside the tropical tree canopy. And the way they get dispersed throughout the tropics is by living in leaves. And then when the leaves fall, they float down to the forest floor. And then those xyleriales fungi, they wake up in the leaf litter and then they go colonize a woods substrate from there. All right. Another exciting part of this whole saprobe life story is the fungal fight club. If you find any dead piece of wood, log, tree, and you cross cut it and you look at the end grain, you will see these colonies of fungi that are fighting for territory. And so these could all be different colonies of the same fungus, or they could be different colonies of different species. And what they're doing is they're all fighting to occupy territory to decompose. And when they meet a competing fungus, they both put up chemical barriers, melanins and different extractives chemicals, because Fungi are chemical magicians, and so they try to wall off the competition and put a wall around their own territory, and that's how they fight for territory. So the Fight Club ecology and the process of succession is really super interesting. And so what happens is the pioneers get a foothold, and then other stuff arrives and they fight for territory, and then they put up their walls and they mark their claim, and then the colonies can be different species or the same species, but different colonies. And what happens is the early colonists are not good defenders and they quickly get replaced by the next wave. So I want to talk about saprobes that change their lifestyle. And this beautiful sulfur shelf, Laetiporus, is a good example of a heart rot fungus that starts as a weak parasite and gradually works its way into the heartwood of a tree. And then once it's in the heartwood, it functions as a saprobe. And I went through that slide before because I wanted to impress upon us all that none of these things are static. All of these nutritional modes that we talk about are in a state of flux and change. And so another thing that happens is if you start paying attention, there's a predictable sequence of colonization. And if you look on the left, you see turkey tail, and turkey tail is an early colonist, a white rot saprobe of hardwoods mostly. And what happens is turkey tail colonizes first, or it's an endophyte, and it starts spreading throughout the deadwood. And then the maize-gill polypore, Trametes betulina, cannot colonize that piece of wood until the turkey tail starts to change the chemistry. So what happens is that the maize-gill, Trametes betulina, is a parasite of the mycelium of the turkey tail. And so the turkey tail is in the wood, it's decomposed in the wood, then the maize gill comes along, it attacks the mycelium of the turkey tail and it kills that mycelium and it takes over. So this is the secondary colonist, this is the pioneer colonist, and this happens throughout the forest with wood decay. Pioneer colonists get taken over and attacked by various secondary colonists. Here is another example. Burkhandura adusta is a pioneer colonist. And then after a while, the lumpy bracket takes over, Trametes gibosa. So this pattern is repeated over and over again in the forest in the drama of wood decay. And so this is sort of a synopsis of what I've just been going on and on about. And so over the course of decay, it could be a couple hundred years, there are definitely predictable waves of saprobes that will come and decompose the wood as the chemistry of the wood is altered and changed by the predecessors. Pretty cool, really, if you think about it. So what happens, how does a fungus uh, find its next meal, basically? So three ways, really. Latent propagules, what we talked about as endophytes, that's one way that a fungus gets to a new meal, new organic substrate. And then what we used to think was the main way, spores landing on a scrape or a wound or a broken limb, that's another way in which a decay fungus gets to a substrate. But the way that's really interesting to me anyway is when the fungus exhausts the nutrients from one substrate and leaves that substrate and crawls underneath the leaf litter or the needle litter and goes searching for a new substrate. And so that's what I want to talk about next is that phenomena. Uh, and so what we can do, if you're a log flipper like me, and you never want mushroom season, fungus season to end, you keep flipping logs until January, you start to notice that mycelium on the underside of logs has particular geometry. And I would call this a fan-shaped geometry right here. And that geometry tells you, and this is from the work of Lynn Bodie, who's been studying this since 1980. She would say that this geometry indicates that this fungus thinks it's in a nutrient rich environment. And so it makes a fan shaped mycelial mat to go out in all directions to find a new substrate as close as possible. So it doesn't have to travel long distance. So that's the architectural uh, strategy for finding near nutrients in a nutrient rich environment. And then conversely, this is an architecture that indicates that the fungus, it's going to leave its substrate that it's been on, but it believes that it's in a nutrient poor environment. And so it's going to have to travel a long distance underneath the leaf litter to look for a new substrate. And so you have these thick cords going out in different directions rather than the thin fans. And so something that happens that's really interesting, and I'm going to go back for a slide just for a minute. If you look at this fan, when this fan goes out into the environment, if it finds say another log to colonize, say right here, this stretches out into the forest. Whoops. It stretches out into the forest here and it finds a log. What'll happen is all the other mycelium will contract and form a cord here. So all this other mycelium here will contract and form a cord here so it can shuttle all its resources to the newly found substrate. And if you're a mushroom grower, you know this and you've probably observed this on your agar plates. And so this is the long distance strategy. All right. I hope I'm not going on too long. We're getting near the final. We're in about the eighth inning now. So one of the reasons I was really interested in studying saprobe ecology after all these years is because for about 10 years I was studying ectomycorrhizal symbiosis and all the ectomycorrhizal fungi. And so I learned from studying ectomycorrhizal fungi that when an ectomycorrhizal fungus wants to produce a mushroom, it doesn't really fruit from the ground. It fruits directly from a colonized tree root. And it uses the energy that the tree supplies the fungus on the colonized root to make a mushroom. But I started to notice ectomycorrhizal fungi in fairy rings. And so I didn't really have an answer for why that was happening. And I kept asking people, why do ectomycorrhizal fungi make fairy rings if they're fruiting from a colonized tree root? And after a long time, I found out that they do not get rid of all the genes that they have that code for decay. They don't get rid of those genes that code for those enzymes that can decompose things like leaf litter or grass or needles. They keep them, but they're just silent until they need them. And what happens with a fairy ring, like this is an Amanita muscaria, I believe. A fairy ring like this is the fungus leaves the colonized tree root, We call those extra radical mycelium when they leave the tree root. And before it forms a mushroom, it runs out of energy. And so it has to switch over to a saprobic mode. And then it acts like a regular decomposer in a lawn. And it spreads out and it forms a fairy ring. All right. So I want to talk about evolution, and that's going to wind up the whole show. Ectomycorrhizal symbiosis came from saprotropic lines 60 independent times, at least. And why it happens so many times is it's fairly easy for a fungus to switch from being a saprobe over to becoming a mycorrhizal mutualist. All it has to happen is two things. That fungus has to lose or quiet down the genes that code for decomposing lignin and cellulose. The loss of the decay fungus function allows that fungus to approach a tree root, and then they gain some genes that code for small signaling molecules that enter the tree root, and they disable the tree's defense response. I want to say that again. Two things have to happen for the evolution of decay fungi to ectomycorrhizal fungi. They have to lose the decay function, and then they have to gain the genes that code for disabling plants' defense mechanism. And that's all it has to happen for a fungus to evolve over millions of years, of course, into an ectomycorrhizal fungi. And it's happened at least 60 independent times. And here's a chart of that. And I borrowed this idea from Francis Martin and David Hibbett, but we're going to end up with this. If you go back 300 million years here, where my cursor is, what you see is sort of the modern version of white rot. And that traces back to the auriculariales, the jelly fungi, were probably the first lineage of modern white rot fungi. Other lineages are much older, 400 million years. And then about 180 million years ago, here on this dotted line, all of these saprobes, white rot saprobes at first, they started changing and evolving and doing what I just described to you. They lost the decay enzymes and they started dialoguing chemically with plants. They started dialoguing chemically with plants. And so you have this progression from ancient saprobe here to like a Galerina, a modern wood decay white rot fungus evolving. I don't know why that keeps happening, but evolving to an ectomycorrhizal. And then over here, you can see in the Amanita lineage on top, you have something like Amanita thersii, which is a saprobic Amanita. So you have an ancient white rot ancestor, and then you have a more modern white rot Amanita, and that evolves into modern Amanita ectomycorrhizal species. And then the brown rot is a special case. The brown rot only happened because of the invention of that Fenton chemistry I was talking about. And the brown rot evolutionary pathway leads to the bolatales. So you have ancient white rot ancestors over here, and that evolves into brown rot, something like Serpula. And then the brown rot lineages, and those are things like Tapenella, Serpula, Coniophora, they evolved into modern boletes again by losing their plant decay enzymes and disabling the defense system of plants. And so to wrap everything up, fungi are mad chemists, and that's really what's so fascinating about them. And their superpower is chemical deconstruction. And as a result of that, we have medicines, we have food, we have nutrients and materials that become new things in forest ecosystems. And they have all these superpowers, and but at the end of the day, uh, it's all a lot of rot. And that really is my program. I'm sure I went on way too long, but I'm happy to listen to questions. If you stuck with me this far, I hope you followed along, and I hope you enjoyed the story I told tonight.
We did. Man, that was awesome, Bill. I'm ready to hear all that all over again so I can recall it a
Uh,
little bit better. I don't see too much in the way of comments, questions, but I did have a couple that I wrote down for you. So you were mostly focusing on basidiomycetes. Do ascomycetes perform rot in the same um, by the same more or less chemical design, or do they have their own thing going on?
Yeah, ascomycetes do use a lot of the same general categories of decay. Ascos are usually early colonists of deadwood and plant material and late stage colonists of deadwood and plant material. And their decay function is somewhat superficial with respect to wood. They do not decay large volumes of wood the way basidiomycetes do, but they do use the same categories of enzymatic tools, which are peroxidases and chemical enzymes that deconstruct polymers. So the basic functionality is the same, but different enzymes.
If anybody has any questions, feel free to just unmute yourself and go ahead. Uh, some
I'm
uh,
surprised,
some compliments coming your way.
I'm surprised that, you've got these things that are creating brown rot. It's got all that pesky cellulose, you know, out of the way. Why wouldn't something come in and be like, oh, wait, this should be easier to break down now and digest? I don't know. You'd think that kind of like how you were talking about, you've got these secondary mushrooms moving in. You'd think after it starts turning to brown rot, it's freed up more of that to be digested by something else. But I guess something hadn't evolved to get that yet, huh?
The thing about it is, Mitch, is with brown rot, what's left over is lignin that's been reconfigured, and it's really,
Ah,
really hard for any fungus to decompose lignin. It's so energy intensive to decompose lignin that the fungus ends up getting no net benefit.
that probably also makes it exclude some other kind of lignin decomposer because now it's changed the lignin. It doesn't even have a chance to get home. Oh, that's interesting.
Yeah, lignin is amazing, and it's used in all kinds of industrial processes, also, and it's widely studied all over the world for the chemicals involved and the fact that it's such a complex molecule. It's really difficult to even imagine how it forms because I am not a biochemist at all, and it took me a long time to even get a rudimentary understanding of this chemistry. But it's so complicated that practically every lignin formed is a little bit different than any other lignin. And there's a whole big controversy about whether or not fungi inventing white rot decay is responsible for the end of the Carboniferous coal deposit phenomena.
I've
Uh,
heard before, yeah.
Yeah, and there's been lots of things written about that. David Hibbett has written about that extensively. The idea that the reason why mostly all the coal in the world was formed during that 60 million year period, 360 million years ago to 300 million years ago, is because before that, the modern form of lignin degrading white rot hadn't been invented yet. Uh, and so those high lignin components of coal happened during that period. And then as fungi invented more efficient lignin decomposition enzymes, that was a contributor in the ending of coal formation. But he got a lot of pushback from coal scientists about that, who insisted that that's not the reason, that the reason the Carboniferous period ended is because the Earth got cooler and the climate dried up. It wasn't so wet, so you didn't have all that anaerobic non-decomposition happening as everything dried up. So it's a good controversy, though. And if you ever bump into David Hibbett, ask him about it because he would love to talk about it. Well, I hope I didn't go on too long. I get involved in the storytelling, and Zoom is such a thing that you can't see the audience really. So much
Boy,
different than giving a live performance.
Go ahead, Chris.
Yeah, Bill, enjoyed the talk. It was excellent. I just had a quick question. I was kind of waiting for you to drop that, $64,000 word, or Groucho Marx would send the duck up on a swing or something. I recall colleagues in grad school working on enzymes called lactases, and they
Yeah.
were particularly interested in lignin. And I did not hear you mention that, so I was just curious if you could address. I haven't followed it. What is the role of lactases, if anything, in the breakdown or buildup of lignin?
Yeah, I mean, this is one of those class 2 peroxidases. And it's one of there's between like 9 and a dozen uh, class 2 peroxidases. Lactase is one of them that works together to break down the bonds in lignin, because lignin is so complicated. It takes a whole enzymatic cocktail to break it down. But I don't know the particulars of the function of lactase.
Great,
But
thank
I
you.
hope that gives you a rough answer anyway.
Yeah,
It's
no, it
part of that.
does.
Those class 2 peroxidases are something that are pretty remarkable as a class of chemicals that fungi have. And it's really interesting. If you look at the 1000 Genome Project, they mapped out, I don't know, hundreds of these class 2 peroxidases. And a typical fungus might have hundreds of copies of genes that code for the same enzyme. So there's a huge redundancy in the genome for these enzymes that code for all these different kinds of decomposition, which really makes you wonder why, why is there so much redundancy? I'm sure there's a good answer that a biochemist could give us. It's probably something to do with tandem repeats and horizontal gene transfer, but I am not a biochemist, so I'm not going to pretend to know the answer to that.
Uh, yeah, I'll just mention it too. I mean, years ago, I haven't heard much about it, but APHIS approved a low lignin pine tree for cultivation down in the southeast in South Carolina, North Carolina. And our first thought was, well, is it just going to fall over, or is it going to get colonized by various fungi? And I have not heard boo about it. I'm often wondering if it was a successful cultivated product.
I'm wondering if that was a short rotation tree. Uh, I mean, lignin is sort of the glue that stiffens up plant cell walls. And so if you have a really tall tree, you need lignin. But if you have a short rotation tree or a tree that never gets too tall, maybe a low lignin content cell wall is not a terrible thing. I don't know.
Yeah.
I don't know
Well,
the
thanks
answer to
again.
that. You're
Yeah.
welcome. That was fun. If anybody ever wants to ask me questions when I give a program like this, I am always open to responding to either emails or messages. I'm like sort of fungal obsessed, so you're not going to be invading my space if you bother me on a Sunday morning or something like that. Although
I
I
have
might.
Bill, could you just, this is Scott Shapiro. I was just wondering if you could repeat the word of the night that you got so excited about at the end of the hyphae. I forgot.
All right, Spitzenkorfer.
Bitzenkorfer.
I recently went
Thank
to
you.
a mushroom festival as the Wizard of Spitzenkorfer. So that's going to be one of my new. Yeah, Megan just wrote it for you.
Oh, thank you. Maybe you can show up for a talent night as Spitzenkorfer.
Well, don't mention that because I will be at Sequinota.
Thank you. That was great.
All right, thanks.
Thanks, Bill.
I did have one more question for you, Bill, and I think that's
Okay.
about it. So I'm wondering, whenever it comes to this, if there's a part of this rotting that is analogous to the chemical erosion that you see with lichens growing on top of rocks. They do create some chemical erosion on rocks that they're attached to, but most of that is because the lichen is secreting waste material, generally in the form of carbolic acid, into
Uh-huh.
the rock itself, and that's breaking down the minerals. I mean, uh, with us, with what you're talking about tonight and breaking down organic molecules, I mean, could some of that be just because the fungi is secreting waste materials and unintentionally doing some of this, or?
Yeah, I don't know the answer to that, Tom. I'm sure it's possible. One thing I did not get into because it's just a big subject is that part of the saprobic lifestyle includes enzymes that mine rocks and minerals mostly for nitrogen and phosphate. Yeah, so that's another part of the decay story is mining minerals with enzymes. Yes. I mean, they're fungi. The story just it blossoms out over and over and over again, and you start bumping into other things like geochemical cycles, right?
Oh, we could easily extend this out to a 2-hour talk, but
I could, yeah.
I think we've exhausted any questions in the chat, and I don't want to hold you online with us any longer. I'm sure we'll have some time between now and Sequinota to come up with some new questions for you whenever we see you next. I want to thank you so much for coming out tonight, Bill. Man, this was awesome. I can't wait to watch it again as soon as we post the recording. Thank you so much. Thanks, everybody. We'll see you on the mushroom trail soon. Bye now.
Good night, everybody.